There's a switch in the back.
Yeah, sure.
It's off.
Right.
So,
So this is our shaker works like. Can you guys see it? So we can adjust the speed.
that's the max speed, I guess.
So then it moves faster. 1 second. Yeah. So this is. Yep. And then we can see around. Oh, so these are all the centrifuges. This is a centrifuge. We'll go on a more detailed lab tour soon. But for now, till people join, we'll just show you around. There's another centrifuge there. Then there are more reagent bottles. These are the powder forms. All of these are the powders. The ones that you saw before were the liquids. And again, more powders. Then this is an incubator. I will talk about this soon. That's a fridge. And then you have a microwave, actually, which is also used in the lab, not for cooking. And you have, this is called an autoclave. It looks like a washing machine, but it's an autoclave. And we have another lab here. And I'll talk about all of these soon. But until then, it's a quick lab tour. Do you guys have any questions? No? No questions at all. Okay. So which classes are you guys from?
I am from 5, going to 6.
I'm from 6th.
Mm-hmm.
I'm from 6th standard.
Nice. Are you going to 7th or are you going to 6th?
I'm in 6. I am going to 6. I am.
I'm going to 7th.
Ooh, nice. Then Devabrata. Did I say your name right? Devabrata.
Yes, ma'am.
Which class are you in?
I'm 8th.
8th? Ooh, nice. And where are you guys from? And Vijaya Malika, you're in which class?
Ma'am, I'm from 8th standard.
Okay. And where are you guys from? Like Kerala?
I'm from Tamil Nadu.
I'm from Kerala.
You're from Kerala?
I'm from Kerala.
Hey,
I'm from Odisha.
sort of from.
You're from?
I'm from Odisha.
Oh, nice.
Where are you from, Orissa, Divya Sai? Divya Sai, that the correct name?
I'm from Kotak.
Cuttack. Oh, wow. We were in Angul for a year. You know Angul? Angul, Simlipada, Angul. Okay.
Yes.
Okay, you guys can unmute, switch on your cameras, not unmute your videos. I learned. But you guys can switch on your cameras if possible. And when are you, have your school started? When is it going to start?
Mine started. Mine will start at 6th June. 6th June.
Ooh, so this Monday. Are you excited?
Yeah, excited. My friends coming.
Ooh, fun.
My school starting at 15th of June.
Oh, that's a long time away. Where are you from in Tamil Nadu?
I'm from Chennai, but currently I'm studying in Bhopal.
In Bhopal?
No,
Yes.
Dubai.
Ooh, okay. I'm from Chennai too.
Me too.
My friend here is from Hyderabad. That helps.
Okay.
We'll wait till like 2:40 and then we can begin. Does that sound okay?
It's already okay.
Did you guys like the cell session?
Yeah.
Yes.
Learn
Interesting about microscope, cells, DNA.
Mhm.
Ah.
Did you all solve the crossword? Oh no, the crossword is in this. Sorry, I will show you the. Yeah,
Oh, I thought.
not crossword, word search.
Yeah.
Do you guys like word searches? We have a word.
Crosswords.
Not
Crossword.
crossword, word search. My bad. We have a word search for you.
Word search.
I'll send the link once we get there. So till then, we shall wait
Yes.
5 more minutes. I can show you guys how a pipette looks like. So this is a very small. Can you get me either 1000 microliter? So these are called tips. And wait a second. Array, it's not in that drawer. Give me a second. Okay. So this is a pipette. Can you see it?
Yeah.
Okay.
What does it do?
Can you see it? It looks like a injection sort of, but it's not one. Yeah? Can you guys see it? So what do you guys think this is used for?
I don't know.
No idea.
Any guess?
Just guess. It's fine.
For injecting something.
Close. Very close, actually. Pretty much that's. Yeah. Debabrata. Sorry. Is it Devabrata or Debabrata?
It's Deva.
Debabrata.
Devabrata. Okay, got it. Yeah. You wanted to say something.
Any guesses?
Is it a dropper?
Correct. Yeah. So it's a very fancy drop.
And then squeeze that. That kind of.
The one you use for the scissor.
Yes. Okay. That's like the. Yeah, correct. Is that the one you guys use for like.
Nose droppers.
Yes. So this is a dropper that we use in lab. So it just
For
has,
leaves. For leaves.
it has some. Yeah. So it has some markings here which show how much volume we're taking, and you can like take it and like release stuff and like yeah. So it's used for transferring things. Ooh. So we have something. Do you know what this is?
Dye or potion.
Yeah, so it's actually just water and it's just mixed with a dye called saffron, and it's actually a
Saffron. Yeah.
saffron in.
Yes.
Okay, so what you're gonna do is like, so you just press the dropper, you insert like put it in, and like you just leave it, and you can suction water like that, and well, like you can transport it to a different place, and then you can release it again, right? So this is how a dropper would work. But for many of our experiments, we need very, very precise, measurements, because this is like, it's kind of approximate, right? You can't really say how much you're taking. So for that, we use a pipette. So a pipette can be of like, you can use it for different volumes and like, yeah, the one that I have right now is a 1 ml pipette, but you can use it for like 0.5 ml or like smaller volumes than that, or if you want to go more than 1 ml, you just need to keep doing it. So these things you can see here, they are tips. So we need a sterile, so in lab, we often need a very sterile environment, right? Everything has to be super clean, correct? Yes, no?
Yes.
Why does it have to, okay, if it's not clean, what will be there? Like, what is the issue? What do you mean by not clean?
Unclean. It's dirty.
Correct. So what causes dirt? Like what is
present in that?
Bacteria.
correct? So a lot of microorganisms are present, which is what you're going to learn about. So you don't want all those microorganisms in your lab area, because you want it to be super clean so that you can grow the things that you want and not like random bacteria or like fungi or whatever it is, right? So you use tip box. tips which can be like discarded. So you can, these are all one use things. So you like press your pipette onto the tip and you
Wait.
give us a second. This tip box isn't very not working very well.
So basically, they are just syringes and syringe needles which can be replaced.
Yeah, pretty much. So this is how it looks. This is how a tip looks. Okay.
So a different volume tip will have a different length.
Okay.
And the size.
So what we're gonna do again is just press down on this. Yeah. Go into whatever you want to suction. Leave it gently. And can you see the liquid's been drawn up?
Yeah.
Yeah. And you can again like release it. See, it's gone. Isn't that cool?
Like, I found it cool.
Yeah, we have never seen. I mean, I'd never seen a pipette before coming to ISET, so it was very like at the first time when I saw it, I was like, oh damn, so cool. So do you guys think it's cool?
It is cool.
Ah, okay. There's one person who thinks it's cool. Hey, everybody else, is it cool or
It
not?
is.
Okay, yeah, awesome.
Yes, it is cool.
Oh, okay. Another cool thing is how we discard this tip. Okay, wait, this is kind of dangerous. I need another speaker. So we have it's like a launching mechanism. Did you guys see that?
Yeah.
So it just goes off like that. So yeah. Okay. So now I think we can start our next session. And I will definitely show you around lab in a more detailed thing, and I'll explain what everything is. So that we'll do at the end of the session. But first we'll go over content for microorganisms. Does that sound good? I guess.
Yeah.
Yes.
Yeah.
Cool. Let's start. Give me a second. No, I need to present my window only. Because it's in my thing. It's not edit access. Okay, wait, but then. Can you guys see my screen?
Yes.
Okay, give me a second.
Yes.
Okay, can you guys still see my screen?
Yes.
Yes,
Okay.
we can.
Okay, so microbiology. So what is microbiology? Do you guys have any idea about it?
Study of microorganisms.
Ooh, very nice.
Yes.
Anything else? There doesn't have to be one definition for anything. You can have several.
The biology of microorganisms.
Yep,
Yes.
correct. Next, anybody else?
The study of microorganisms.
Correct. Okay, so essentially microbiology is made up of two words, right? Micro and biology. Biology is the study of anything that's living, pretty much. And micro is, what is micro? What does micro mean?
Small.
Yeah.
A micro means very small.
Tiny.
Yes.
1 second. Hi guys, sorry. Yeah. So okay, so so some of you answered what is micro. Micro is something which is very, very small, right? So essentially anything that we can view under a microscope. So I think all of you saw what a microscope is in the previous session.
Yeah, section 1.
Yeah. So anything that is seen under a microscope can be called as a microorganism. So microbiology is essentially the study of anything that can be viewed under a microscope. Does that make sense?
Yes.
Yeah.
So okay, let's go on to the next slide. Okay. So now you know what microbiology is. So when you look around you, okay, when you look at your hands, when you're looking at the air that you're breathing in, or the water or soil, what do you think is it? Do you think there are organisms in all of that? Anybody?
Simplifying the question, do you know where
Can you repeat?
are present?
Yeah. So there's water around you, there's soil, there's air. Do you think there are microorganisms present in all of that?
Yes, ma'am.
There's microorganisms present in soil.
Do you think there are microorganisms present right now in my hands?
Yeah.
Yes, there are bacteria and
There
microbials.
are tons of microorganisms in, everywhere, even in the air.
Correct. So microorganisms are present all around us at all points of time. In fact, a very, very cool thing is there are a lot of microorganisms which are present inside of us right now in our intestine, and that is called as the microbiome. Okay, so there are actually more number of microorganisms inside you than your own cells. Can you guys imagine that? So there are, I think, close to 300 million microorganisms inside your intestine at any point of time. So is that scary? Or is it good?
Ma'am, there are good bacteria and bad bacteria also.
Correct. So exactly. So like what Dibya Sai said, microorganisms can be classified into two types. You can have good microorganisms which help us a lot, and we won't be able to do
Bad microorganisms.
Yeah, we won't be able to do a lot of things around us. And there are also bad microorganisms which cause a lot of diseases, and they're not good, right? So yeah, and there are also organisms in the middle which don't do anything. They're not good, they're not bad, they just live. So there are also organisms like that. And they are called free living microorganisms. Okay, so these are the different types of microorganisms that are present. And here there's a small video about how microorganisms move around. And yeah, you can see this is under a microscope. Okay, we're looking at this under. So it looks super big, right? But like to the naked eye, you won't be able to see anything, right?
What are these?
So
These are microorganisms.
Yep.
these are a class of microorganisms. They are rod-shaped, right?
Yeah.
Yeah, what are they
Yeah.
moving like worms?
So they are not moving by themselves because the water or whatever they are placed on is moving. So that makes the microorganism move.
But microorganisms can also move by themselves. That's
Yes.
also possible. Okay, so moving on. So yeah, like I said, right, in the lab, everything has to be super clean and like sterile, right? Why do you think that is? So why do you think that is? So what if it's dirty, right? Like we are walking around, it's fine. Like we're living in our houses, we don't care about like, oh no, are there microorganisms everywhere? We don't think about it. We're like, yeah, sure. Like as long as it doesn't affect us, we're good. But in the lab, we're very, very specific about these things. So we wear gloves, we work inside a lab. I'll talk about that soon. We make sure everything is sterile. Like we clean everything. We wipe it down with ethanol, which is like so it kills all microorganisms. So why do you think it's so important that we work in an environment which doesn't have microbes?
One more question. So follow-up to that question to make you better understand it. So you all have visited doctors, right? So they collect your blood samples sometimes. Before collecting your blood samples, they they alcohol wash it. So with a small cotton or this thing, they clean the area where they're going to take
Where
your
they're going to
blood
prick.
from.
Yeah. Why do
So
they do that?
why do you think they do that?
So when they take blood, microorganisms will
Okay.
also
One person. Rohit, go ahead.
the microorganisms will enter the blood also as well.
Okay. And why do we work in a very clean environment in lab?
Now, because if anybody gets the in contact and gets exchange of microorganisms, nobody knows if they are good or bad.
Correct. Okay. And GB, Vinu? Go ahead, answer.
Yes, GB.
Wait, are you saying something? Because I can't hear you.
Yeah, he's saying something.
Is your audio? Wait, can other people hear him, or is it just me?
You can't hear him.
Are you saying it? Okay, can you type it in the chat box? Is that okay? Cool. Eric, did you have something to say?
No.
Oh, okay. Cool. So yeah, both of your points are very valid. One is like, you don't want microorganisms to enter your blood, or you don't want the microorganisms to enter the sample that is being collected. And here, when we're working in lab, we also work with microorganisms. We grow microorganisms in lab. So there are two reasons why we don't want other microorganisms to be present. One, or like it has to be clean. One is for our own safety. So we don't want to be like, there are like Debabrata said, there are many organisms which are harmful to us, right? So we don't want to be inhaling it, or we don't want them on our hands when we're like, if we will wash our hands before we leave lab, but there's always a chance that we could carry it and we could potentially ingest it, or it could cause some harmful effects. So we don't want that. One. Two is we will be growing certain microorganisms, right? We don't want other microorganisms to grow instead of them. Does that make sense?
Yes.
Okay. So how do we? Yeah. Hasini, did you want to say something?
No, nothing.
Okay. Cool. So how do we grow microorganisms? So in the environment, you can see that microorganisms pretty much grow on anything. Like water you give, they'll grow. Soil, they'll grow. Like air, they grow. It's fine. They have nutrition and everything. But in lab, like I said, everything is sterile. It's not a very conducive environment for them to grow. Like it doesn't help them grow, right? So we need to give them something for them to grow. So we need to give them nutrition. What else do we need to give them? Guys, anybody? Nutrition is important. Then what else is important?
Proteins, energy,
Correct. All of that comes under nutrition. Yes, no?
vitamins.
Ma'am, darker place and small, most place.
Sorry?
Ma'am, darker place and most place.
I cannot hear you very well. That could? Okay. So, yeah. So someone said temperature. Who said temperature? Rohit, did you say temperature?
Not, I didn't say temperature.
Okay. Do you think temperature is important?
I think temperature is.
Do you think we can dark and wet place? Yes, dark and wet place is very important for like outside. Correct. You can see fungi growing. You can see algae growing in dark and wet places.
Fungus?
Sometimes, yeah. Did you guys say something?
Fungus?
Fungus can also be microbes. So an acid or
A hot liquid.
yeah. So
Yes.
an acid is something which makes the environment like acid. So it makes it acidic. It makes it like
Bonds.
correct. So there are a lot of. Do you guys know what hydrogen is?
Hydrogen, yeah.
Yeah.
So,
Yeah, it is a gas.
okay, for a more simpler explanation, so you drink water, right? So water's pH level is close to neutral. So your body is able to withstand that. That pH is what your body needs. pH is basically measuring how
acidic a substance is.
Yeah.
So you have any substance can be classified into three. It can be acidic, it can be neutral, it can be basic. And this acidic, neutral, basic classification comes from measuring the pH. So it's like, say, measuring your height. Okay, you can say people are short, you can say people are like normal height or like common height, then you can say people are tall, right? So how do you do that? You do that by measuring the height of the person, correct? So similarly, by measuring how acidic a substance is, which is called as pH, just P and H, right? You can say whether the substance is acidic, it can be neutral, or it can be basic.
just like how you cannot drink salt water.
Yep. So I'll give you an example of something which is acid. So what do you think a lemon is? Do you think lemon is acidic, neutral, or basic?
Acidic.
Yep.
Acidic.
A lemon is acidic. So if you like try tasting lemon, it's super sour, right? That is because of the acids present in lemon.
Oh.
And for a base,
Citric acid.
yes, citric acid is the acid which is present in lemon. Very good. Then when you know the soap water that you use to wash your hands.
Yeah.
Soap acid.
Alkali.
No.
That is a very good example of a base. So that's something which is basic. Yeah. Okay. So essentially what I was trying to say here is that growing of these microbes need very, very specific conditions. So they need specific temperatures. They need specific, like someone said, like sunlight exposure or like no sunlight. Some of them need oxygen to grow. Some don't need oxygen to grow. So all of them need nutrition to grow. Like you need, like Rohit said, there's proteins, there's carbohydrates, there's fat. All of it is important for microbes to grow. So we need to supply these things, right? Because we are growing them. So we need to give them food. Like just how like your parents are giving you guys food every day, we need to give our microbes food to make them grow, correct?
So a proper sufficient amount of food for them to grow in a controlled manner, so that we can measure their growth and make sure they are like in constant number, and we can use that.
So the food that we give is called LB, or it's named after the person who made it for the first time, and that's Luria Bertani. And this is how it. So this is how it looks in real life.
So can you guys see this?
The yellow color liquid.
Yes, sir.
Yeah.
So this is full of nutrients.
Nutrients for the bacteria.
Yeah, not for us.
Yes.
Then what will happen if we drink it?
If we drink it, we've never tried it. We don't recommend anybody to try it. But yeah, so it has so it has something called as LB powder, which has tryptone, yeast extract. So all of those are essentially proteins. Okay, so it's a very nutritious medium. So we call this a media. Can you guys say it? Media.
A nutrient medium.
Okay, so this is the liquid form. Okay, but this liquid form isn't always very helpful for us to grow, because it's always going to be the bacteria is also going to be in liquid, right? We don't want that. Sometimes we want to, how we eat from plates, we have our food in our plate, just like that bacteria can have their food in plates. And these are,
so these are the nutrient plates for
we
them
can't
to.
see it, huh?
Can you see this?
So it's like a jelly. So what we do is we take.
See, over here you can see there's a lining, right? Over here.
Can you see it? Petri dish.
Yes.
Yes, this is
not a petri.
this is not exactly a petri dish. It's a plate, but yeah, it's
Yeah,
close.
so it's a bigger dish, petri
dish is usually smaller. This is slightly bigger.
And it's open, so we can close this. Over here we have a lid, so we close this over here, and as you can see, this is where our bacteria grows when we solidify it.
Okay, so what we do, how we make those plates? Essentially, we take this liquid, right? And we add something called agar to it. Does anybody know what agar is? No? Nobody knows what agar is.
It's a jello.
No.
Okay. Have you guys had jelly?
Yes.
No.
Yes.
Rose, you never had jelly.
Yes,
You don't
ma'am.
like-
I have heard jelly, but I have never eaten it.
Okay. So yeah, so you guys have you guys made jelly?
No.
Yes.
Gelatin and water.
Ma'am, to
freeze it.
make jelly, we need gelatin.
Gelatin.
Or you use have you guys seen those ready-made packets? Like you just add it to water and you like boil it and
Gelatin.
Yeah, that is
Gelatin
gelatin.
powder put, then
so it
water.
can either be it can
Gelatin put, then water, then freeze it. Jelly.
you don't freeze it, you boil it and then you freeze it. So gelatin is one option. The other option is agar. So agar is just like another setting agent. So you guys seem to be familiar with gelatin, right? So agar is derived from algae. So it's like a vegetarian option for gelatin because gelatin, do you know where gelatin is derived from? Okay. It's derived from pigs. Yeah, bones of pigs. Yes, correct.
Oh,
And
I don't know.
okay, Pranav did not know that. So
Yeah.
Pranav's also learning with you guys.
Yes.
So that is how gelatin is derived, but agar is derived from algae. So you actually can get agar from supermarket. So next time you guys go to a supermarket, you can ask your parents to see if there is agar around, and it's actually available because people use it for cooking a lot. So what we do is we take this liquid broth, we add agar to it, and we set it like a jelly. And if we set it like a jelly, we get this plate like this. It has the media inside. Can you see it? Okay, so then what we do is, so then we grow our bacteria on top of it. So we take we have like stock bacteria and we put it on the LV agar, and because it has all the nutrients in it, it will start growing. Sounds good?
Yes.
Okay,
Yes,
so
ma'am.
next.
Why do you need to grow microorganisms?
So we use microorganisms for a lot of our experiments. So you guys, we study them. We study their growth. We can extract a lot of useful things from them. And we make them make proteins for us. We make them like, you guys will learn about vaccines.
Yeah, vaccine. Yeah.
Yeah.
Isn't it?
I mean, they can be used to make vaccines, yes.
So basically, what you call it, when you want to experiment something, you cannot experiment it on humans, right? You cannot experiment it on your species. So sometimes these single-celled organisms mimic a multicellular system. So to study what happens in that basic controlled environment, you can extrapolate it to a multicellular organism. Am I making sense? I don't think so. I'm making sense.
Okay, so essentially what you're doing is like you're taking this organism and you're doing your studies with this small bacteria. Then you get some interesting results with it, right? Then you can go ahead and like move on to other models. That's what we call them. We call them model systems, and you can even eventually come to like using that on humans. So it's very cool. So it's like this hierarchy that you follow. Okay, so microorganisms can be of different types. So one thing that you guys have constantly like mentioned or I've heard is bacteria, but bacteria aren't the only thing which are like under microorganisms. You can have several things. You can have fungi, you can have protozoans, archaea, viruses. All of them come under microorganisms. So this is just like a pictorial representation of like microorganisms in general. So you can see that they're different in shape, they're different in size, they're different in color, they're different in structure. Yeah.
And you can see they use different structures to move around.
Yeah. All these, these lines that you see, all of that is used to move around. Right? Okay. Yeah. So these are the different types of microbes. So you have bacteria, you have archaea, you have fungi, you have protists, and you have viruses. out of this, how many have you guys heard about? Have you guys heard about bacteria? If you guys have heard about bacteria, click on the raise hand button.
Yeah, we had.
Okay, a lot of people have heard about bacteria. That's awesome. We love bacteria here. We work with in our project. We work with bacteria. So yeah, we are very excited about bacteria. Then, have you guys heard about archaea?
No.
Who's heard about archaea?
No.
If anybody's heard about archaea, raise your hand. Oh, so Jimmy,
Actually, the most of the time we have only heard about bacteria, fungi, and virus.
correct. Okay.
Yeah.
So, yeah.
Ma'am, I have heard about archaea, but I have in our next
Time,
time, we have not actually started it that much, so that's why I don't really know.
That's fine. We'll give you guys a brief intro about each of it. Okay. So yeah, like you guys said, the most common types of microbes are bacteria, fungi, viruses. This is what most people know about. But then, and study. But there are other types of microbes as well. Okay. So yeah, we'll go to the next. So first up is bacteria. So bacteria are single-celled organisms, and
It's a water bottle. It's an enclosed water bottle.
okay. Yeah, sorry. I wanted water and I asked Pranav to get me water, and he's getting me a very random water bottle from somewhere. So I refuse to drink water from it. So bacteria. Okay, wait. Why did I refuse to drink water from it?
Anyone. Brownie points for anyone who answers.
Yeah. Why did I not want to drink water from
Because in that it can be bacterias.
No, it is water. It is water.
Then
Yeah,
what's
but-
the thing
yeah.
you might refuse then?
Bhatesh, yeah, you are going on the right track. Because there could be
Ma'am, can I repeat the question?
because
Or
there could,
dirty water.
yeah, there could be microbes in the water, and I did not want to drink that. So yeah. Anyways, so now moving on to bacteria. So these are single-celled organisms, and they have different sizes and shapes and colors. And one very cool thing about So one interesting thing is do you guys know about unicellularity and multicellularity?
Yes.
Okay. So what are we? Are we unicellular or are we multicellular?
Multi.
We're multicellular. So
Multicellular.
what does that mean? What does it mean to be multicellular?
Means in our body
Ma'am,
there are
1%.
One or more
Can you
cells.
guys use that raise hand thing?
It has more cells.
Okay, Hasni, go ahead.
Well, we have more than one cell.
Okay. Bhatesh, that's right.
yeah.
Yeah, you raised your hand.
Yeah, so that only I was
You are
telling.
also going to say that.
Yeah,
Eric,
same.
were you also going to say the same thing or something else?
Our cells, our cells breaks and we are different cells. One cell breaks into different cells.
Okay, I mean, close. But essentially, yeah, like multicellularity just means that we're made up of several different cells. So we have different types of cells in one organism. So can you guys name some of the cells that we have?
Ma'am, red blood cells, nerve cells, fat cells.
Wow, very good. That's awesome. Correct. So we have all of...
Brown cells.
Sorry?
We even have the liver cells, muscle cells.
Super.
Yes.
Awesome. Great job. So there are different types of cells, right? So why do you think we have different types of cells? Immune cells,
For
yeah.
different types of work, ma'am, because one cell,
Because every cell is adapted or
Ma'am, cells work for different
cells
types of
that work
purposes.
for the whole body.
Correct. So different cells perform different functions. So they do different work. And because they do different work, they all work together. And finally, we have the human body, which is an entire organism which can function by itself, right? But one very cool thing about bacteria is you have just one single cell which performs all these different functions. So you have, nutrition, then you have digestion, then you have breathing, then you have excretion, right? You have thinking. All this is done by different cells in our body. You have transport, right? But all of these things are done by one cell in bacteria. So that's very cool.
But
right?
not with the same level of
Yeah, it's definitely not with the same level of complexity. Like, we are super complex organisms. We have evolved and we can do a lot of complicated things. Bacteria are very, very simple organisms, but it is still cool that they can do all these different things with just one cell, right? Then you have, okay, so again, you have good bacteria and you have bad bacteria. One good bacteria that we like almost, I really like curd. Do you guys like curd? Like milk, curd.
Cheese.
Not cheese, but cheese is...
But curd. But
yeah, curd.
it's the most common thing in every household.
Do you guys like curd?
And yeast.
Yeah.
Who and all like curd?
And yeast.
Yeast. Yeah, yeast. Will come to yeast. Hold on a second. Okay. A lot of people like curd. I approve, guys. I also really like curd. So this curd is what? That's curd.
Yeah, we plated.
Where is it in the middle? Okay. So this curd is actually made using bacteria. So so how do you make curd? So you take milk, then you take a little bit of curd and you add the curd to the milk, and then the milk becomes curd. So why do you think we add that little bit of curd to the milk?
Because when we add curd, it's my theory. I guess that it will happen. I guess
The curd has the bacteria and the bacteria helps it become to the curd. When it's
Okay, so 1 second. Can you raise your hands and then we'll go in turns? Because then it... Okay.
Divya, say first.
Yeah, Divya Sai.
Yes, curd has the bacteria that helps the milk to convert into the curd.
Very good. So that's the answer pretty much. And Bhatesh, if you want to add something to it, yes.
Yeah, so it is my theory. I'm guessing that it will be like that. So when we add that little bit of curd, in the bacteria in that grows up and makes all that milk into curd.
Correct. So why we're adding that little bit of curd is because that little bit of curd has a lot of bacteria, and that bacteria will essentially, you know what it does? It produces acid. So this acid helps in the milk to become curd. You guys now know what an acid is, right? So it produces something called lactic acid, which will convert the milk to curd. And you know what the bacteria is called? Do you know what the curd making bacteria is called? No? Any guess?
No.
Fine. Okay. It's called Lactobacillus. And we actually took some curd and we grew it.
We plated it yesterday. So, okay, sorry.
We grew it.
We grew it yesterday. If you can see can you see dotted lines on dotted spots on it? I don't know.
Yes.
No.
I could. I can.
Wait. Let me try it at this angle. Can you see different spots on it?
Yeah.
Yes.
So white.
Yes.
Yeah, white spots, right? So,
Yeah.
so this white. Oh, Gopal is telling me to turn off the torch, but I cannot show it without turning off the torch. Can you see white spots on it?
Yes.
Yes.
So these spots are the bacteria. The bacteria which is present in the curd. It's called Lactobacillus.
Okay. Yeah. So bacteria can be very helpful to us, but you can also have some diseases which are caused by bacteria, like cholera, dysentery, all of that is caused by bacteria. And one interesting thing that I actually learned only yesterday is that the smell that is, that you can smell after rains is actually caused by bacteria. I didn't know that till like 2 days ago. Did you guys know that?
Excuse me, ma'am.
Yeah.
Yes.
Ma'am, what is
No,
the
after rain, that I was saying that because of water and soil get mixed, that why that smell comes.
Yes. The bacteria present in the soil releases that odor, that smell.
Ma'am, what is the difference between bacteria and viruses?
We'll get to
Okay,
it.
there's a lot of difference. I will teach you what bacteria is, then I'll go over what virus is, then you can tell me what the difference is. Okay? Sounds good?
Sounds good.
Ma'am.
Awesome.
Virus is smaller than bacteria.
Yes, but wait, we'll get to viruses. Virus is something which is now super talked about, so I'm sure all of you are excited to know about it, but we will get to viruses. Let's first go to archaea. So, from what I saw, a lot of you guys don't know what archaea is, or like you've never heard about it, and I'd never heard about it till I think like 10th or 11th, so it's not surprising at all. So archaea are actually like super old bacteria. So these bacteria are like some of the first living things that were present on Earth. So Earth was formed 4.5 billion years ago, right? And archaea were actually some of the first organisms to start living in Earth. So they are around like 4 billion years ago, like 4 billion
So
years
how
ago.
the archaea were made means
What?
How would
how
they
the archaea.
Oh, how do they come into existence? So, okay, so that's a little complicated, but essentially what happened was first there were some like proteins which were just present, like which just, proteins are just nothing but like carbon, oxygen, and hydrogen, right? All of life is made up of mainly these three elements, right? So proteins were made, and these proteins, they, there was always sunlight was there, so there's a lot of UV and like radiation.
very harsh environment?
Yeah,
at that point.
and there was also a lot of now we have a lot of like nitrogen and oxygen in our atmosphere, but at that point of time you had a lot of other gases. You had methane, you had oxygen, you had nitrogen, you had ammonia. So a lot of gases were there, and there was lightning and like a lot of. It was a very hostile. It was a very like it wasn't very.
It was not a place where you could live properly.
Okay,
So
it was
you
a very
could-
harsh environment. Okay. So what happened was, so there was this UV radiation and stuff. So these elements started like coming together to form molecules, and these molecules came together to form macromolecules or bigger molecules, which are proteins and carbohydrates. Then these proteins and carbohydrates came together to form these archaea, and from archaea the process of evolution started. Like archaea like grew, evolved, like just how we evolved from apes. Archaea, like bacteria, evolved from archaea. So yeah.
So like how the temperature of the earth and everything, it was in hostile climate. From hostile climate, it slowly it became into a cooler environment. So
Means archaea are the ancestors.
Yeah, archaea are the ancestors, exactly.
Yeah.
You can actually easily remember that, like A and A, like archaea ancestors. So yeah, so these are super old, like bacteria almost, but you don't call them bacteria, you call them archaea. And so
What's the difference? Means like
so they're just much more simpler. They haven't evolved as much as bacteria. Like bacteria themselves are simple organisms. Archaea are even more simpler.
oh.
Okay, and they're just like super. It's like saying, what is the difference between humans and like apes?
Oh.
Right. So they are more simpler organisms than us. Like we can do more complicated things. We have evolved a little more. So just like that. Bacteria have evolved from archaea, right? So these archaea, like I said, so they were growing in very harsh environments. So even now they can grow in very harsh environments. So like we need oxygen to survive, right? Do you think all organisms need oxygen to survive?
No.
No. Okay. Yes, that's correct. So not all organisms need oxygen to survive. Organisms that need oxygen to survive are called aerobes. Okay, can you guys say that? Do you guys know that?
No.
Okay. So they're called aerobes, and then you have organisms which can survive without oxygen. So any wild guesses as to what they would be called? It's the opposite of aerobes. Just like English, aerobes.
Non-aerobes.
very close. Non-aerobes, exactly, but we call them anaerobes. So aerobes and anaerobes, okay? Anaerobes are organisms which don't need oxygen to live. So a lot of bacteria, archaea, all of them are anaerobes. So they don't need oxygen to live. And which is again very cool. So they can survive in places where we cannot survive. So these archaea, they can grow in very harsh environments because that's where they grew from. That's where they evolved from. So they can grow in places which is very, very salty, like saline. They can grow in places which are super hot, like 100 degrees Celsius or higher. That's when like water boils, right? But then you can have archaea living there. Then you can have archaea living in very acidic places. Okay, now you guys know what acidic is again. So like in acidic places, archaea can grow. So these are all places where not a lot of organisms can grow, but these organisms can grow. So they're special that way, right? Next up, we have fungi. So not all fungi are classified as microbes because like you guys said, microbiology is the study of something which you can visualize under a microscope, right? But are all fungi microscopic? Can you guys give me an example of a non-microscopic fungi?
We have fungi at our door.
Oh.
Like
Yeah,
mushroom.
There are wet places, no?
Correct.
There. We can see with our eyes.
Correct.
But naked eye.
Great answer.
Naked.
Mushroom is a non-microscopic fungi. So they're like very big, right? You can see them with the naked eye. So they're not microbes, but they are fungi. But you also have fungi which are microbes, like yeast. Okay. So do you guys know what yeast is? Have you heard about yeast?
Yes.
Okay.
Which goes on in the crust of pizza.
Yes.
Raise hands.
Zero.
Can you guys raise your hand and then answer? I mean, use that button. It grows in bread.
Yes.
No.
It grows in yeast, rust.
Okay, Eric, answer. What is yeast?
It's bacteria we used to make pizza.
Very good.
Bread is used to make yeast.
Because
bread
of air,
yeast or
no,
is yeast
the
used to-
bread gets very fluffy and air goes in there.
Correct. Hasni, yes. Hasni, do you want to answer? Okay. So yeast is actually used for making bread or like pizza dough, like someone said. So yeast is a fungi. So you take your flour, you add water, you add yeast. And what this yeast does is it produces carbon dioxide. It produces a lot of carbon dioxide when you give it nutrition to grow. So a little bit of yeast you'll add and then it will become a lot of yeast. And all of this yeast will produce a lot of carbon dioxide. And because it produces a lot of carbon dioxide, the whole like dough will like rise. Okay, it will poof up. So if you have like this much of dough in the beginning, you'll have this much of dough towards like say in a couple of hours, right? So after that, that's why like when you take bread, you can see holes in breads, right? That's because of the carbon dioxide which is produced by yeast, which when baked, it'll be holes. Got it?
Even in cheese.
Yeah, even in cheese. So so their walls contain a protein called chitin, which is unique to fungi. And they also don't have chlorophyll, so they cannot perform photosynthesis. So several of these microbes, some of them are photosynthetic. So what do you mean by photosynthetic?
You make food out of sunlight.
Correct.
Yes.
So you use sunlight.
Sunlight, nutrients present in the soil,
Correct. So you
And
take
water.
and
And water also.
carbon dioxide.
and water. Yes. So you take UV from the sunlight and you use that to make food from like again carbon, hydrogen, and oxygen, right? And that so these are called photosynthetic organisms, and there are several photosynthetic organisms like plants, and you also have microbes which are photosynthetic like cyanobacteria. Okay, but you also have several microbes which are not photosynthetic, so they cannot but we need like raw ingredients. We can't make those raw ingredients, right? So just like that, there are other organisms which can't make their own food. So they depend upon other organisms for their food. So fungi is a class of organisms like that. So they can't make their own food. They depend upon other organisms for their food. Okay. So this is okay. So many of you talked about fungi and bread. So yeast is useful fungi and it's used to like make bread. But there's also fungi which can grow on bread. So when this is actually an experiment that you guys can do.
Yeah, actually,
bread mold.
actually I've done, instead of bread, I have taken that chapati.
Okay. And you've done this experiment of growing fungi.
Yeah, I've put in that into 4 days in an airtight container.
Okay.
Yes. And what did you observe?
So I observed that little black spots were there.
Yes.
And bread mold.
Correct.
Yes.
Bread mold. So bread mold is the common name. So the actual name of the fungi that grows is called Rhizopus. So what if you guys haven't tried out, some of you have, which is very nice. But if you haven't, you can take a slice of bread and you can damp it a little bit with a little bit of water. Don't add too much of water, just a little bit of water. And you keep it in like a airtight container and keep it in a dark place. And within like 4 or 5 days, you can see some green, black stuff growing on it. And that's fungi. Make sure you don't eat it or touch it. And make sure your parents know that you're doing it. Okay.
Yes. And if you touch it, wash your hands properly.
And we'll actually show you guys proof as to why you guys should wash your hands. Okay. Okay. So next thing is protists. So again, I don't think a lot of y'all knew about protists, and that's again totally understandable. It's not something which is very talked about. So protists are again unicellular, and you have two plant-like protists and animal-like protists. The plant-like protists will be able to perform
Photosynthesis.
Yes.
Good job. And the animal-like protists won't be able to perform photosynthesis. And one example of, do you guys know what malaria is?
Malaria?
Mm-hmm.
Yeah.
Yeah.
Okay. So what do you think causes malaria?
Mosquitoes.
Correct.
Mosquitoes.
Yeah. So this is something that a lot of people think is that mosquitoes, gives rise to malaria, which is actually wrong, because it's not the mosquito itself which produces malaria. It's the organism that the mosquito carries, which is called a plasmodium, which is a protist that causes malaria. So mosquito is actually very innocent in all this. So like supposing I have malaria, I've been infected with something called a plasmodium, which is a protist. Okay. So that is creating all my symptoms and it's making me sick. Okay. So the mosquito, when it bites me, what happens is it carries the plasmodium from my blood and then say it comes and bites Pranav. So the mosquito again is not doing anything. It just is transporting this plasmodium from my blood to his blood. So once it transports it to his blood, he also gets infected with malaria. So that's how it works. So the mosquito is called a vector and it doesn't actually cause malaria. What causes malaria? What it is?
So
Organism causes malaria.
Plasmodium. Can you guys say that again?
Plasmodium.
Actually, the worst part of biology is their names.
True.
So basically, if you want to understand it more simply, so imagine this is the protist, okay? So, and this is our mosquito. So what happens is mosquito sucks in the blood. Along with the blood, there is protist. So now this mosquito is carrying the blood. And the blood is being carried and like it needs to drink a lot more blood, right? The mosquito. So it goes to a different host. From there, by mistake, the protist enters our bloodstream. So basically, protists have a dual life cycle. Like, should I explain life cycle? No. Yeah.
Okay.
So it needs a particular host to grow in. So, and this acts as a courier guy.
Yeah. So malaria is caused by protists. Then, so one fun fact is that you have some protists which cause infections in the intestine. And in fact, they have 2 nuclei. You guys would have learned about nucleus in the cell class, right? Yeah. So they have 2 nuclei, so it kind of looks like they have a face because you have 2 nuclei and the protist itself. Okay, so this is Plasmodium that causes malaria under the microscope, and another protista. Finally, we have viruses. So I'm sure all of you know about virus. Yes.
Yes, ma'am. Unlike
Coronavirus.
Yes. So COVID has made a huge impact on us in the past couple of years. And correct. There are also other viruses. Do you guys know about other viruses?
Omicron.
Sorry?
Omicron.
Yeah, Omicron. All of that is under coronavirus. So it's coronavirus that is split into several subtypes.
Like bird flu.
Sorry?
Bird flu.
Yeah, bird flu. Like
Swine flu.
you have Ebola, you have Nipah, you have so you have like H1N1. So there are a lot of different viruses, but the one that we all are very, very, very familiar with is COVID or coronavirus, right? So these, okay. So
the HIV virus.
Correct, HIV virus. Yeah, very good. So do you think viruses have life?
I have heard that sometimes not, but sometimes.
Ma'am, because they are also microorganisms, and microorganisms are known as living organisms. So that's why it is also a living.
That's a very good argument. Good job. Then everybody else, what do you guys think? Do you think viruses are living or non-living?
Both.
Ma'am, they start living.
We start living once we enter a cell of
we can
correct. Very good.
Yes, ma'am. We can define if they are non-living or living.
Yeah. So essentially, when viruses are very, 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 very small. Okay, they're smaller than bacteria. They're smallest organisms that I can't say they're the smallest organisms that are existing. But they are very small. Okay, you need like special microscopes. So you guys saw some microscopes in the morning, right? You can't see viruses under that. They're too small for that. So you need more powerful microscopes to see viruses, right? So so the other thing is, so viruses are non-living if they are not inside an organism. So they can be in the air around me and they are non-living. They're like a dust particle. They don't do anything. Okay, but the issue is once they enter an organism, in the case of COVID, it's humans, right? So viruses enter humans, and then what they do is they take over the mechanism of our cell. There are viruses that infect viruses. Ooh, interesting. I know about viruses which infect bacteria. I don't know about viruses which infect viruses. If you have any examples, do let me know. So yeah, so once you have viruses which are coming inside our body, they take over the mechanism of your cells. So they take over virophages. Good job. So yeah, so you take over the mechanism of the cell and they make the cells produce more viruses. So they make the cells produce some proteins, they make the cell produce some RNA and DNA, which you learn about in tomorrow's class, and they make more viruses. So essentially outside the cell, they're fine. They're like a particle. They don't have any life. They're all good. Once they come inside the organism, they take over the mechanism of my cell. And they make my cell produce all these things and produce more viruses. And from me, I now become like a factory, right? I create more and more and more viruses. And these viruses can go on to infect other people. So this is why they're very dangerous, right? Because not only, so it's very Okay, so now I'll give you guys a question. So it's very hard to develop treatments for viral infections. Why do you think that is?
If it is new to us, then it will kind of take 5 or 6 months to even study them.
To catch a virus to test it out.
Yeah, sorry, go ahead, Eric.
Because you have to catch a virus to test it out.
Okay. Devabrata?
Can you repeat the question?
Okay, so it's very hard to develop, any treatments for viral infections. So you don't have a lot of medicines for viral infections. Why do you think that is?
And because we need to take precautions.
We need to what? They are not living. Yes, kind of. Okay, all of you guys gave me good points.
Ma'am, I think it is because mostly the medicines are actually made up of the disease or virus, whatever it is. Because if a medicine for a specific disease or virus is made, we need a little bit of its own substance or whatever, so that we could make the medicine. And mostly some of the viruses or diseases are not that much available, and we can't find them. So that's why it is probably a little bit hard to
Okay, but we can have viruses in labs. We can work with viruses in labs. So that's not an issue. We'll be having viruses and we can use them to you'll have some amount of virus to study, work on and everything.
Ma'am, maybe because the viruses are present inside our cells, so the vaccine will even affect our cells ourselves.
Very good. That's that's pretty much, yeah. Yes, go ahead.
Streptokinase is a bacteria or a virus?
What? Streptokinase. Streptokinase. Streptococcus is a bacteria. Streptokinase is an enzyme.
Okay, ma'am.
Okay, Streptococcus is a bacteria though. Streptokinase is just like a protein. Okay, so so what I think Vijaya said was very close. So they take over the mechanism of our cell, right? So to produce more viruses. So anything which stops the process will affect our cells. So we will be taking medicines which are affecting our own cells, which is not good, right?
Yes, ma'am.
Which is why it's very difficult to develop therapies for viral infections, right? So then you have bacteriophages, which are actually viruses which infect bacteria. So not only can viruses infect big organisms like humans, you can have viruses which infect bacteria. This is an view of coronavirus under a microscope. So this is how it looks. Okay, so now I think it was Debabrata. Didn't you ask me what was the difference between bacteria and virus?
Yes, ma'am.
Okay, now can you answer it? Hello? Okay. Are you there? Can you hear me?
Yes.
Okay. Okay, cool. Debabrata, if you can answer it at some point, that will be awesome. And we will move on to the final. We did a little experiment for you guys. So why do you think we need to wash our hands?
Ma'am,
Because our hands consist of
because our hands are
bad bacteria.
microorganisms
Okay, raise your hand.
which are unaware of, and we don't know if they are bad or good.
Asni, go ahead.
To get rid of the germs on our hands.
Very good. Anybody else has anything different to that? If you do, you can jump in and say.
To remove the unwanted bacteria from our hands.
Correct. Then the BSI.
Actually, I have heard that 99% bacteria are good for ourselves and only 1% are bad.
Okay, I don't know if that statistics is correct, but a lot of bacteria are good for ourselves, but I don't know if it's 99% of bacteria.
Yeah, I mean, most of the.
Yeah.
And viruses are most of the harmful.
Yes. Okay. So yeah. So essentially, why we need to wash our hands. So now I'll show you guys a little experiment that we did.
Ma'am, I have read that there are kind of 27 types of microorganisms on our hands.
27 times?
Types.
Yeah. There can be a lot of different types. That's great. Fun fact. I didn't know. I don't know the statistics, so it's great that you guys can tell me this. I'm learning as well. So, yeah. So this is what we did. So we took a plate like this containing what? What does this have? What did we guys taught? We taught you, right?
Bacteria.
No. Okay. This has bacteria, but like what is
Food source.
correct? What is a food called? What is a food called? Yeah, Vijaya, go ahead.
No, ma'am, I raised hand for the previous question.
Do you guys remember what it's called? No?
And the question is not
Agar. So this thing has something called as Agar, Luria Batani Agar, right? So this is a food source. So what we did was we just took our hands without washing them and we just kept them on top of the plate like that and we removed it. So what do you think happened? Anybody?
Can you repeat the question?
Okay, so we took...
Can you please repeat the question?
Yeah. So we took a plate which has agar, which is a food source for bacteria and other microbes. And we didn't wash our hands and we just took 4 fingers and we kept it on top of the plate, like inside the agar, like on top of the agar. So what do you think happened?
The bacteria grew.
When those germs will come in our hand.
No, there are no germs in that. This is created in a very
No, the microorganisms will go to the plate.
Correct. So microorganisms which are present in our hands went onto the plate, and when they went onto the plate, they started growing there because they have a lot of nutrition, right? So they can grow. So which is what you can see in plates 1 and 2. Can you see this plate and this plate? Can you see the white spots, brown spots? All of these are microorganisms which are growing, mostly bacteria. Yeah? Can you guys see it?
Yes.
Okay.
And due to getting the favorable environment, they started growing.
Correct. So then what we did was we washed our hands nicely with soap and then we again pressed it on top of agar, and that is the last plate here. Can you guys see this?
Yeah, now it is clear.
You can see that there are not as many bacteria.
Yes.
Now
and there are kind of 4.
Yeah, there is still a few bacteria, that's true, but most of the bacteria are in there. So do you guys now realize how important it is to wash our hands? Because even though we cannot see any of the microbes on our hands, every time, like if we try to if we give them enough conditions to grow, they're going to grow. So there are a lot of bacteria, some of which can be harmful to you on your hands at all times. So it's very, very important that you guys wash your hands well with soap right before you eat. Yes?
Yeah.
Good job.
Yes.
This is something which is very theoretical, right? We have all seen the Dettol ad, the Lifebuoy ad, which is like, wash your hands with soap, then, it'll become clean. But you never had any proof. Now you have proof which says that you have a lot of bacteria before and much less bacteria after. So it's very important. Okay. So now you guys can go for a 15-minute break and then we'll have a small activity. Yeah?
So do we have to join or means we have to leave or?
No, you can stay here. And we'll resume the session at 4 o'clock. So till then, if you guys want to go ahead, go somewhere, like, I don't know, eat something and come back, we can do that. Or if you want to sit here and have a chat, that also we can do.
Okay.
Ma'am, actually, I worked with the microscope.
You work with a microscope?
Yes, ma'am.
Oh, nice. What do you see under the microscope?
And so when do we have to
One
come back? Mama, do we
Ma'am,
have
one
to
time
go?
I saw kind of on a glass, there was written small intestine. So I checked out in the microscope, it was kind of pink and purple color.
Oh, very nice. Is this in school?
But the microscopes are very, very
Yes.
expensive.
Yeah. All of the lab equipment are very expensive. Do you guys
I
want to
just-
in particular in lab?
Ma'am, we have to now meet on 4 or what?
Yeah, you
I
guys
just-
can come back at 4 o'clock.
I just come with the Google that microscope and I go into the shopping. The prices are like 87,000, 32,000, 1 lakh and 3-4
Yeah.
lakhs.
This one pipette costs like 10,000 or 12,000.
Oh,
My God.
Yeah, everything in lab is very expensive. So you should take a lot of care for all of them. You to make sure you
Ma'am,
handle-
every Sunday I go to the lab and work with the electronics and lab things.
Ooh, very nice. Very interesting. What do you guys want to do? Do you know what you want to do in the future?
Scientist,
Oh,
yeah, scientist in ISRO or NASA.
scientist and astronaut and doctor.
scientist and?
Astronaut and doctor.
Oh, very varied. What about
Actually,
others?
means from my childhood, I have a curiosity that whether aliens are there or not. So
Uh-huh.
because
Yeah,
of
there are
that
aliens.
curiosity, I am going in NASA or ISRO.
Valid, fair enough.
I think there are aliens.
There are actually interesting research going on with this debate, whether aliens are there or not. Space biologists.
Astrobiology, that's what it's called.
Astrobiology.
You guys can look it up.
Oh,
I don't know a lot about astrobiology.
actually, I have a very, very big expectation with James Webb Telescope.
Ooh,
Nice.
Because it means scientists have worked very
I
hard
also
and
saw
it
the
is
moon with the
more than
telescope.
1 billion. So obvious you
Is
have
this-
very high expectations.
my break. As in?
What?
And do you guys want to see anything in particular in lab? I can show you and explain.
Yes.
What do you want to
Yes.
see? Is this our break? It's your
Have
break.
you're talking about that machine which we can see viruses?
Okay, that we don't have here. Because again, that's very, very expensive.
We have
I mean, we don't have it in this lab.
lab, yeah.
Yeah.
So
Because
we
it's
have
very-
a separate lab working on it. So they fully work only on
because
that.
for viruses, it's very important that you have to handle it with even more care because they're very small and they can